Application of magnetic nanoparticles. Under this topic is cell separation. Due to the advantages of size and shape, magnetic nanoparticles have a lot of applications in biomedicine, such as cell separation, drug carriers to cure cancer, and hyperthermia. The size of nanoparticles gives them the advantage of going into the body and can be divided into the small scale to cure disease. The following section will focus on the cell separation and drug carrier function of nanoparticles. Cell separation, also commonly referred to as cell isolation or cell sorting, is a process to isolate one or more specific cell populations from a heterogeneous mixture of cells. Drug delivery refers to the technology utilized to present the drug to the desired body site or drug release and absorption. Next. So this is a picture of cell separation. The standard methods of magnetic separation. So you can see the magnet and remove supernatant to fluid flow, then fluid flow, remove magnetic field, so on. With this kind of theory, it can be seen that it can be used in many parts of biomedical applications. It can separate rare tumor cells from blood and can be used to target low number cells. So it can lead to enhanced detection of materials in blood and process technology for polymerase chain reactions. Also, for separation applications, it can be used in optical sensing due to the high sensitivity of magnetic nanoparticles. If the target material is the solid matrix, it can act as the surface to allocate the target material and increase the concentration. Next is the drug delivery. Compared to traditional chemotherapies, magnetic nanoparticles have the advantage of treating the target area more specifically. Due to the guide of the magnet, it can be delivered to the specific site and then release the drug. Since the drug is used only for the specific site, it can reduce the dose amount so that it will reduce the effect of systematic side effect on the body. Next, topical review. The figure shows the mechanism of drug delivery application of magnetic nanoparticles. To further understand the application of nanoparticles and cell separations, I have a video here.
Hi all, welcome back to Nanotech. Today's topic is magnetic nanoparticles can burn cancer cells. We know that cancer has become one of the major public health concerns in our modern society. Magnetic hyperthermia treatment is one of the promising non-invasive approaches for thermal activation therapy on cancerous tumors. Upon exposure to external alternating current magnetic fields, magnetic nanoparticles can generate heat through the oscillation of their magnetic moments. Magnetic hyperthermia can then kill cancer cells by elevating their temperature to 40 degrees Celsius 45 degrees Celsius with minimal injury to normal cells. Magnetic hyperthermia-based heating systems could be employed either for the controlled release of a drug from caged drug delivery systems or the killing of tumor cells or both. The most used materials for magnetic hyperthermia are ferrite nanoparticles, in particular magnetite Fe3O4 or magamite. The size of the magnetic nanoparticle in the range of 10 to 100 nanometer. Magnetic iron oxide nanoparticles have the advantage of long-term chemical stability and biocompatibility, and ease of surface modification and functionalization when compared to other types of magnetic susceptible materials such as certain metals, iron, nickel or cobalt, or metal alloys. Heat generation mechanisms of magnetic nanoparticles in response to an alternating magnetic field. When exposed to an alternating magnetic field, magnetic iron oxide nanoparticles produce heat via two main mechanisms. One is hysteresis loss and another is relaxational losses. Hysteresis losses. Hysteresis losses occur in large magnetic iron oxide nanoparticles which possess multiple magnetic domains. When such particles are subjected to an alternating magnetic field, the orientation of the magnetic moments will align continuously with the direction of the magnetic field. This results in a difference in energy that released in the form of heat. When magnetic iron oxide nanoparticles size decreases, the number of magnetic domains will also decrease until a single magnetic domain remains at a threshold size of approximately 128 nanometers. Below this size, magnetic iron oxide nanoparticles are deemed superparamagnetic, and in the presence of an alternative magnetic field, heat is mainly produced by Néel relaxation and Brownian relaxation. Néel relaxation refers to rapid changes in the particle's magnetic moment when exposed to alternative magnetic field. The rapid realignment is opposed by the particle's crystalline structure, resulting in heat generation. Next is Brownian relaxation, which refers to the frictional heat generated from the physical rotation of particles within a supporting medium when the particles attempt to realign themselves with the changing magnetic field. This results the formation of heat. Hyperthermia can lead to cell death via modulation of various cellular processes. One of the disadvantages is the lack of specificity toward malignant cells compared to healthy tissue. Systemic administration of targeted magnetic iron oxide nanoparticles has the potential to improve the specificity of hyperthermia and improve its efficacy. Future preclinical studies should focus on designing magnetic iron oxide nanoparticles that can target and heat tumors more effectively. Various hyperthermia enhancers should be evaluated in combination with magnetic hyperthermia, with the ultimate objective of achieving clinical feasibility. Thank you for watching.